Now we begin our today's class. This is another operon which is involved in other type of metabolic pathways. Earlier we have seen lac operon, and this lac operon, it is particularly involved in catabolic pathway. And this lac operon, it is controlled by two different manners: positive regulation as well as negative regulation. Now, next operon that we have to see is the trip operon. Now, this trip operon, it is another kind of operon which is present in E. coli, and this operon, it is involved in synthesizing. It is involved in synthesizing a particular type of product. Means this operon, it is involved in biosynthetic pathway. Now trip means tryptophan. Tryptophan operon. This is the another operon which is present in E. coli. Now as you know that in a protein, these proteins, they are synthesized from 20 different types of amino acids. And these 20 different types of amino acids, they have been either synthesized or they have been taken outside from the environment or from the medium. Now in case of E. coli, this E. coli, this bacteria is possibly, it has the ability to synthesize all the 20 different types of amino acids by its own. Without using external amino acid, it can synthesize its own amino acid which is required during the protein synthesis. But the amino acids which are required during the protein synthesis, they are required in very large amount. And so this bacteria, whenever it is possible to take it from the environment, it takes from the environment. And if these amino acids, they have been not available in the environment condition, then the operons which are involved in synthesizing these amino acids, these 20 amino acids, they have been expressed. And whenever the concentration of these amino acid is low outside as well as inside the bacterial cell, in such situation, all the operons which are involved in synthesizing these 20 amino acids, they have been expressed. And when this concentration of these amino acid is low, then it is expressed. But when it is present in higher amount, large amount, in such situations, these operons which are needed for the synthesizing these 20 amino acids, they have been shut down. Okay, when these amino acids are present in tremendous amount, at this situation, these operons, they have been shut down. But whenever there is a requirement, at that time, these operons, they have been expressed. Now we have to see one of the example in E. coli, that is a trip operon which is involved in synthesizing the amino acid tryptophan. And this amino acid is synthesized from a substrate that is calcimate. It is a substrate from which the tryptophan it is synthesized. And for synthesizing this tryptophan from calcimate, it requires 6 different enzymes. And these 6 different enzymes, they have been synthesized by 5 genes. Here, one gene produces an enzyme which functions in two steps. In this tryptophan biosynthetic pathway, there are 5 genes products they have been required in order to synthesize the amino acid tryptophan inside the E. coli from calcimate. Here, one enzyme functions in 2 steps. Now this tryptophan operon, you can see this. This is the tryptophan operon. Here is the regulatory gene. That regulatory gene, it is called as a TRPR. That is TRP repressor. It is having a promoter. Okay. Then this is the original tryptophan operon, which is needed for the synthesizing the amino acid tryptophan. And these 5 genes are there, which produces 5 different proteins, 5 different enzymes, and these enzymes, they have been participating in order to synthesize this tryptophan amino acid from carcimate. Now you can see here, this is a P-trip. It is a promoter from tryptophan operon. This is the operator. Then comes the another sequence that is known as a ladder sequence. And now how this tryptophan operon, it is regulated. Here this TRP repressor, it synthesizes a mRNA by a transcription process. From that transcription process, that mRNA, TRPR mRNA, it is translated to form a inactive repressor. That inactive repressor may also called as aporepressor because it is inactive form. Now when it get activated, when the amino acid, tryptophan amino acid concentration goes very high, in such condition, this tryptophan act as a co-repressor. It get bound to this aporepressor, which is present in inactive form. Now this aporepressor plus tryptophan, which is acting as a co-repressor, these two get combined and they form the active repressor. Now this active repressor, it get bound to the operator of that TRP operon. When this condition occurs, when there is a high level of tryptophan is either by synthesized by this TRP operon, or they may present in a surrounding environmental conditions. In these two conditions, this TRP operon, it is regulated by this TRP repressor, which is already, when it is synthesized, this repressor is synthesized, it is present in inactive form. But only it get activated when it combined with amino acid tryptophan. Means this TRP operon, it is controlled by the tryptophan amino acid. If the amino acid concentration, tryptophan amino acid concentration is high, then tryptophan act as a co-repressor. It get bound to the aporepressor and they form the active repressor. This active repressor then get bind to the operator of that TRP operon. When it get bound to the TRP operator side of that TRP operon, then this RNA polymerase enzyme, it is not capable of binding to the promoter side, because this promoter and the operator side, they are having overlapping each other. Then in such conditions, this RNA polymerase enzyme, holoenzyme, it is blocked from transcribing the TRP operon. And when this TRP operon is shut down, when the amount of tryptophan it is present in the cell, it is in high amount. Again, when the amount of this tryptophan get lowered, it get decreased, then this tryptophan, it get released from this repressor, it get released from the operator. Then this RNA polymerase capable of accessing this promoter site, get bound to the promoter and starts transcribing this TRP operon genes. When the tryptophan amino acid concentration is low. But when it is present in high amount, this tryptophan combined with aporepressor forms the active repressor, and this active repressor bounds to the operator and stops the binding of RNA polymerase in order to initiate the transcription of this trip operon. So this trip operon, it is controlled in this manner by tryptophan itself. Now this trip operon, it is also controlled. This is the same figure. This is a regulator. It is from the inactive repressor, and this inactive repressor is inactive stage when the tryptophan level is low. In such conditions, these genes, they are expressed, these proteins, these enzymes, then participates in synthesizing the tryptophan amino acid from glycine. And again, when the concentration of this tryptophan amino acid it get increased, then tryptophan it get bound to the aporepressor, where here this tryptophan acts as a co-repressor. It bonds to the aporepressor. It forms the active repressor complex. Now this active repressor complex get bound to the operator site of the trip operon. When it get bound to this site, then RNA polymerase enzyme is not able to access this promoter site. It is not able to bind to this promoter site and to initiate the transcription process. So that's like this. The tryptophan itself controls its own synthesis. This is the mechanism through which this tryptophan is operated in an equalized cell, and it is regulated in equalized cell. Then you can observe this in this video.
The tryptophan operon, TRIP operon for short, is a bacterial system for regulating the synthesis of the amino acid tryptophan. The operon controls expression of 5 genes. These structural genes are transcribed when RNA polymerase can bind the promoter controlling them. But when the cell has lots of tryptophan, why waste energy making more? The TRIP operon is at one position on the circular bacterial chromosome, and a regulatory gene for the system is about a quarter of the way around the chromosome from it. This gene, called TRIP-R, encodes a dimeric repressor protein on a slow but ongoing basis, and the repressors diffuse through the cytoplasm. We'll see in a little bit how the repressors work. Now let's look at the control sequences for the TRIP operon. They include a promoter, the binding site for RNA polymerase, an operator, which overlaps the promoter, and a leader gene, TRIP-L, which has an attenuator sequence which we'll also look at soon. When the bacterial cell's tryptophan levels are low, RNA polymerase can bind the promoter and transcribe the structural genes. Each gene encodes a protein chain for an enzyme in the TRIP synthesis pathway. TRIP-E and D for anthranilate synthase, TRIP-C for indole glycerol phosphate synthase, and TRIP-B and A for tryptophan synthase. As tryptophan is synthesized, it diffuses through the cytoplasm. When it encounters a TRIP repressor, it binds a site on the repressor, changing that protein shape. The repressor can now bind the operator, block polymerase binding, and prevent transcription of the structural genes. Thus, tryptophan is said to be a co-repressor. Like the lac repressor, the TRIP repressor does not stick like glue, but periodically dissociates, allowing a low, basal rate of transcription. The operon can fine-tune tryptophan synthesis. In essence, it can read various levels of tryptophan concentration in the cell.
So,
tryptophan
like this, the TRIP operon, it is regulated by the TRIP repressor and protein. Now, similarly, like that of lac operon, this TRIP operon, it is also regulated by another layer of regulation. And this another level of same, in this case also, this TRIP operon, it is also regulated by another way. Now, this regulation also takes place at the transcription initiation level that we have seen in earlier case, that is TRIP repressor case. Now, similarly, this TRIP operon, it is also controlled at the transcription elongation phase. When it is controlled at when it is regulated at transcription elongation phase. That controlled mechanism is known as attenuation mechanism. And this additional level of control mechanism, it helps the bacteria to adjust to the environmental conditions. If there is an increase in amount of tryptophan, this helps to delicately balance the amount of amino acid tryptophan to that equalized set. When the concentration get increased, it repressor protein is functioning there. But if repressor protein is not active, in such condition, RNA polymerase begins the transcription. Now this attenuation control mechanism, it is controlled by a specific sequence which is present in that structural gene of that, which is present. You can see here, this is the trip promoter, trip operator, and this is the ladder sequence where the attenuation mechanism has to control the transcription process here. Now you can see here, when this trip operon, it is regulated at the transcription elongation level, that mechanism we call it as a attenuation. And this transcription term is terminated in a particular region. That region we call it as a ladder region. And how much time this attenuation takes place, it is decided by the amount of tryptophan which is available. Means how many times this attenuation mechanism exists there, it is dependent upon the concentration or the availability of the tryptophan in the bacteria. And this attenuation mechanism is controlled through a sequence which is present in the ladder sequence. And this ladder sequence, which is having near about 162 nucleotide pair bases, which is present at the 5' end of the mRNA and preceding to the initiation codon of the first gene. You can see this is the mRNA which is produced from this trip operon, and this is the leader sequence which is made up of 4 different regions which are indicated in different colors. This is the one region 1, this is the region 2, this is the region 3, and this is the region 4. And when this mRNA is transcribed, from the structural genes of the trip operon, in such condition, this ladder sequence, which is made up of these 4 regions, they are capable of binding to each other to form stem and loop structures. Okay, when the amount of tryptophan is low, at that time RNA polymerase enzyme binds to the promoter, then initiate the transcription process. And as it initiates the transcription process, it is followed by the ribosome, which again start the translation process simultaneously. As the mRNA is formed, same time the ribosome, it get bound to the form mRNA, and from that mRNA molecule, it starts translating the mRNA to form the protein. So it is a simultaneous sequence in the bacteria. Transcription process, it is followed by translation process. As the mRNA is transcribed, at the same time ribosome also carry out the translation to form the mRNA, from that mRNA to protein. Now when the leader sequence is transcribed, this leader sequence contains near about 162 nucleotides, and it is made up of 4 different regions. As you see here, region 1, region 2, region 3, region 4. These 4 regions, they play important role in attenuation mechanism of the trip operon. Now, as this leader sequence is transcribed, these four regions, they are capable of forming stem and loop structures. When this region 1 and region 2, they are paired, they are forming the secondary structure. That structure is called as a pause loop. And when this pairing of first region and the second region of leader sequence occurs, when the pairing of this first and second region of the leader sequence, leader mRNA occurs, it forms a secondary structure. That structure is called as a pause loop, and where the RNA polymerase pauses and slow downs. Similarly, followed by this pause loop, there is another secondary structure is formed. That secondary structure is called as a terminator loop, which is formed by pairing of third region and the fourth region. Here you can see that when first region of the leader mRNA get paired with the second region, it forms the pause loop. So because of that, this pause loop, the RNA polymerase enzyme, it slows down. Similarly, there is another secondary structure is formed, and that secondary structure is formed between the region 3rd of leader mRNA and the region 4 of the leader mRNA. And this loop which is formed, that loop is known as a terminator loop. And followed by this terminator loop, there is a presence of poly-U sequence. Poly-U sequence. So this poly-U sequence, it also plays a very important role in controlling the attenuation mechanism. Now another loop structure is formed. That loop structure is formed when region 2 it gets paired with region 3. When region 2 pairs with region 3, that loop form, or that secondary structure which is formed there, that secondary structure is called as anti-terminator loop. Okay, that second structure, it is formed, it is known as anti-terminator loop, and this plays a very important role here. When this pairing of region 2 and 3 occurs, it forms the anti-terminator loop, and when this region is paired, at that time then transcription takes place to the structural genes. When this region 2 and 3 they get paired, then RNA polymerase enzyme continues to transcribe the structural genes, that is E. Then next one, that 5 genes which are present in that structural gene of trip operon, they have been transcribed when there is a pairing of second region of leader mRNA and the third region of leader mRNA. When this pairing takes place, this forms the anti-terminator loop. So this is the basic understanding of the leader sequence. Here the leader sequence of that mRNA which is transcribed, it plays a very important role in order to attenuate this trip operon. And this pairing of this 4 region decides whether that structural genes of trip operon they have been transcribed or not. Now this process occurs in a simultaneous manner. This transcription process is followed by translation process. Immediately it is followed by the translation process, and this translation process and transcription process, this finally adjusts according to the level of tryptophan. Now here in region 1, it is found that there are 2 tryptophan codons are there on the leader mRNA region 1. Usually this tryptophan amino acid, it is normally present in E. coli 1 for 100 amino acids in E. coli protein. But in case of trip operon, they are present in 2 numbers. Here in this case, the leader sequence which is transcribed, it is also translated. It is also translated. Similarly, but this happens not in every cases, but it happens only in trip operon, where the leader sequence it is transcribed as well as translated. And during the translation of the first region, there is the requirement of 2 tryptophan amino acids. Why? Because there is a presence of 2 tryptophan codons in region 1. And this region 1 2 tryptophan codon adjusts. This helps to finally adjust the requirement of tryptophan in the cell. If high concentration is there, low concentration is there, according to that, the transcription and translation process, they helps to control the trip operon. If suppose trip operon works in low tryptophan level, when the level of tryptophan is very low, in such condition, when the leader sequence is to be translated, firstly the region first is transcribed. Okay, firstly the mRNA is formed. From that, when mRNA is formed, when there is a low level of tryptophan amino acid is there. When low level of tryptophan amino acid is there, repressor it get failed from the operator. RNA polymerase enzyme easily bounds to the promoter, initiate the transcription process. As it starts the transcription process, it comes to the leader sequence. Leader sequence 4 regions they have been transcribed, followed by the translation of this mRNA is carried out by the ribosome. As it comes to the region first of the leader sequence, to translate this leader sequence, it requires 2 tryptophan amino acid. Okay, because this leader sequence first region of the leader sequence, it carries 2 tryptophan codons for 2 tryptophan amino acid. If the amount of tryptophan is low, then the ribosome it gets slowed down there. Whenever ribosomes it slows there because tryptophan is not available. Tryptophan carrying tRNA, they are not there in order to fill the A site of that elongation phase. When the tryptophan get available, only this translation process continues, and this translation process it slows here. While it slows there, the RNA polymerase enzyme it transcribes the leader sequence. Presence of ribosome on the region 1 prevents the pairing of region 1 to 2. And as you know that when there is a pairing of region first of leader mRNA with the second region of the leader mRNA, this causes the pausing of RNA polymerase enzyme. As the ribosome is present in the first region in order to wait for the tryptophan to come at the elongation phase of that translation process, it waits there for the tryptophan tRNAs which carry this tryptophan amino acid. So because of that, the region first of leader mRNA is not capable of pairing with region 2 of that mRNA. So there will be no pausing of RNA polymerase takes place. So RNA polymerase continues to transcribe the next region, that is third region. When the third region is formed, the second region of this letter mRNA it is free. It pairs with the third region of the letter mRNA, and there is a pairing between the region 2 and the region 3 of letter mRNA, and this is the anti-terminator loop. This is the anti-terminator loop because anti-terminator loop means here the transcription process will not be terminated. transcription process continues. And because of the pairing of these two region and the third region of the leader mRNA, there is no formation of another secondary structure, that is pairing of third region of leader mRNA to the fourth region of leader mRNA, which is a terminator loop. This is not formed because there is a pairing of second region to the third region of leader mRNA, which is an anti-terminator loop, and because of that there is no termination occurs at this step. And this happens when there is a low level of tryptophan is there. Okay, when low level of tryptophan is there, in such conditions the transcription process continues. But in opposite case, when there is a large amount of tryptophan is there, in such condition, RNA polymerase and ribosomes continues to the leader region. Okay, leader region. Then there is a pairing of region 1 and 2 as the transcription process. RNA polymerase enzyme continues to transcribe the leader sequence. Similarly, the ribosome, it continues to translate. So because of that, there is a secondary structure, stem and loop structure is found in region 1 and region 2 of mRNA. And because of the formation of this pause loop, RNA polymerase enzyme then slows down. And because of the slowing of this RNA polymerase enzyme, as it slowly transcribes the next region, 3 region and 4 region. And when this 3 and 4th region of leader mRNA is formed, it pairs with one another and forms the terminator loop. And as it forms the terminator loop, as you know that this following the 3 and 4 region, there is a presence of poly-U sequence. And this poly-U sequence, it pairs with adenine, and this is the weakest bond which is to be broken. So this bond between the uracil and the adenine, it is easily broken. RNA polymerase enzyme gets separated from this. RNA polymerase enzyme gets separated from the DNA template. And in region 1 and 2, there is also presence of a stop codon, UGA. This is the stop codon which is present in region 1 and 2. And because of this stop codon, the termination of translation takes place in region 1 and 2. So this is how the transcription of the trip operon, it is also controlled at the elongation level also. And this is controlled by the attenuation mechanism, and this attenuation mechanism is only possible because of the presence of 4 regions in the leader sequence of the mRNA. When the concentration of tryptophan is low, it slows down the process of translation, and because of the slowing of the translation process at the region 1 and 2, at the region 1, there is a pairing between the region 2 and 3 of the that is leader mRNA. And so because of that, it forms the anti-terminator loop. And so similarly, this transition process continues to the structural genes, and there is a formation of tryptophan synthesizing enzymes. You can see here, when the amount of tryptophan is low, you can see here these are the 2 codons which is present in the region 1st, and this is the RNA polymerase enzyme. When the low amount of tryptophan is low, this translation process, it stops at this position. They are waiting for the tRNAs which carries the tryptophan amino acid. So because of that, this is the leader sequence which is transcribed by this RNA polymerase enzyme. It forms the pairing between 2 region and the 3rd region of the leader sequence of mRNA. And this is the anti-terminator loop. which is formed there. As the amino acids they are available, because the amino acid level, tryptophan amino acid is very, very low. So the tRNAs which carrying them, they are also searching. They have to take that at the site of protein synthesis of this trip operon. When they get available, they bounce here. Translation process continues. In the meanwhile, this RNA polymerase enzyme continues to transcribe the remaining structural genes of the trip operon. When this happens, when the low level of tryptophan is there. But if suppose the tryptophan level is very, very high, when the tryptophan level is very, very high, here you can see here translation process continues, and because of that there is a formation of pairing between the third region of the leader sequence of mRNA and the fourth region of leader sequence of mRNA. There is a pairing, and when this third region and the fourth region they are paired, they forming the stem and loop structure that is known as a terminator loop, where the termination of transcription process occurs. RNA polymerase enzyme get free. Similarly, in the region 2, there is a presence of stop codon is there, where the translation process is stopped there. And this happens when the concentration of tryptophan is very, very high. You can see this in this video also.
The trip operon in E. coli contains 5 structural genes corresponding to enzymes that convert chorismate into tryptophan. The TRPE gene contains a 5' untranslated region, or UTR, that plays an important role in the regulatory mechanism called attenuation. The 5' UTR contains 4 regions. Region 1 encodes a short peptide containing tryptophan amino acids. When tryptophan levels are high, region 3 pairs with region 4. This structure terminates or attenuates transcription. When tryptophan levels are low, region 2 pairs with region 3. This structure does not terminate transcription. Let's examine how these alternate structures arise. RNA polymerase begins transcribing the DNA. Closely following RNA polymerase, a ribosome begins to translate region 1. When tryptophan is abundant, the ribosome doesn't slow down at the tryptophan codons. By the time region 3 is transcribed, the ribosome has already moved past the tryptophan codons to partly cover region 2. Because region 3 is prevented from pairing with region 2, it pairs with region 4 instead to produce the attenuator, which terminates transcription. The structural genes are not transcribed, and therefore no additional tryptophan is synthesized. When tryptophan is scarce, the ribosome stalls at the tryptophan codons. Therefore, region 2 is not covered when region 3 is transcribed, allowing regions 2 and 3 to form a hairpin that does not terminate transcription. Because region 3 is paired with region 2, it cannot pair with region 4 and form an attenuator. Transcription continues. RNA polymerase transcribes the structural genes, which are translated into enzymes, which in turn synthesize more tryptophan. You can mimic the effect of tryptophan levels by determining how long the ribosome pauses at the trip codons.